DNA is the basis of everything. It's the molecular instructions for life. DNA is the future of medicine, whether it's fighting cancer, neurodegenerative disease, infectious disease. The more that we learn about how DNA can give instructions, how DNA can make cells that do different jobs, the more we'll be able to understand how to give those instructions to cells that are not working properly. I'm Helen Horton, and I'm the Chief Research Officer at Touchlight. I've been in the field for more than 25 years, working on things like HIV and hepatitis B vaccines. I've always been at the forefront of genetic medicines research. Genetic medicine is any therapy or vaccine that has DNA at its source. mRNA, gene therapy, cell therapy, all of these require DNA. I don't think many people had really heard about mRNA vaccines before the pandemic. The reality is they've been around for decades. What happened made people realize that it's these kinds of medicines that we need because they're so fast to produce. Gene therapy and in particular vaccines are there to instruct your immune system. And you can think of your immune system like an army. It's there to protect you. Gene therapy is about giving instructions to the army. The generals will tell the captains what the problem is. Is it an infection? Is it a cancer cell? So they'll tell them what the problem is, they'll also tell them how to kill it. Both my parents died of cancer, so I'm very passionate about finding remedies for cancer. What we need in cancer therapy are very specific medicines so that we're only targeting the tumor and not targeting anything else. That kind of precision is what genetic medicine can do. I think the potential is enormous. There is a big problem with capacity. Genetic vaccines require DNA as a starting material. And the traditional ways of making DNA are just not fast enough to produce the amount of DNA that's required.
Every genetic medicine relies on DNA in some form or other. We make DNA using enzymes rather than using bacterial cells. And what that affords us is to make DNA much more quickly, much more cleanly, and also much more scalably as well. So it may give us an opportunity to address this bottleneck. Doggy bone DNA is simply a piece of DNA in a test tube. You can think about it as an isolated gene that can be used in a lot of different contexts. The reason doggy bone DNA is different is that what we have is a very clean, very small piece of DNA, and the other type of DNA is plasmid DNA that contains not only the gene that you want, but also some other pieces of DNA, such as antibiotic resistance genes. So at Touchlight, we have the benefit of using enzymes which are extremely precise and extremely efficient. We make DNA using these enzymes really, really quickly with extremely high scale, and crucially, with a higher purity than you can make DNA today using plasmid DNA.
I have a lot of hopes for genetic medicine. I think some are maybe a bit aspirational, but what I would love to see in my lifetime is that we can really make personalized cancer vaccines to help people be able to fight their tumors without all of the other side effects. And so that's why technologies like ours, we hope, will revolutionize the field by being able to make DNA quickly in the amounts and at the capacity that we need to.